Hello and welcome back. Hopefully you have finished your organelle test and all of the assignments that went with that unit. So you should have a pretty good idea of what organelles are, these little discrete packages inside of eukaryotic cells that carry out certain functions. And those organelles are going to work together to help cells carry out their life processes. Now, organelles come together to make up cells, and that's what we're going to be focusing on in this unit. So in the cells unit, we're going to look at how certain single-celled organisms have adaptations to help them survive in their environment. And we're going to look at how cells reproduce and how multicellular organisms use certain cells and processes to reproduce. And we're also going to look at how multicellular organisms with all these different types of cells that do different things in the body, how those multicellular organisms get those different cells by using the same template of DNA in every cell in that body. We'll start with unicellular organisms, and uni means one. So these are organisms that carry out all of their life processes in a single cell. They don't need other cells to help them to survive. So these organisms might live together in a colony, but you can take a cell out of that colony that's unicellular and it'll be just fine on its own, and maybe even make its own new colony. Now that's different from a multicellular organism like us that's made up of many different types of cells. We can't take a single cell out of our body and expect it to live just fine on its own. All of our cells are going to have to work together to help us to survive. And that's true for any multicellular organism. So unicellular organisms can belong to any of the three domains, and we're going to look at those here in just a second, but those domains are bacteria, archaea, and eukarya. And they do have some very distinct characteristics, so they're very different from one another. And we're going to study 4 unicellular organisms in this unit: the bacteria, which you see here, the paramecium, the euglena, and the amoeba. So the bacterial cell we've already talked about a little bit, so I'm not going to go too in detail with it in this unit, but we'll give you a little bit of a review here. Bacteria have this outer covering called a capsule, and in that capsule they're going to have these little structures called pili, and pili are like little hairs that help them to stick to surfaces. Bacteria also usually have a flagella, and that's going to help them to move. It's a long tail, and a lot of bacteria have more than one of these. So there's different types of bacteria that can be classified based on how many flagella they have and where those flagella are on the cell. Inside of this capsule, we have a cell wall, and that cell wall is made of peptidoglycan. We'll talk about that more in just a minute. And then inside of that cell wall, we have the membrane, and all cells are going to have this membrane. The inside of a cell is pretty simple. We have cytoplasm and we have DNA. And this DNA is a double helix that connects one end to the other. So it's a circular piece of DNA. It's a circular chromosome. And they have these other little circular pieces of DNA called plasmids that carry certain types of genes. And these plasmids can be dropped by one bacteria and picked up by another, and then that bacteria will have the gene that's carried on that plasmid. So that can be really dangerous with medicine if that plasmid is carrying something like a resistance gene to antibiotics, and that another bacteria can pick up that resistance. So bacteria are very simple cells, and that's pretty much all you need to know about bacterial adaptations. So the 3 domains of life are bacteria, archaea, and eukarya. And bacteria are prokaryotes, and so are archaea. So what this means, pro means before, and karyo means nucleus. So these organisms evolved before the nucleus. And so their DNA is not contained in that nucleus. It's just floating around in the cytoplasm. Now, bacteria, they have a cell wall made of peptidoglycan. I mentioned that a minute ago. And peptidoglycan is just a sugar that is very tough and often targeted by antibiotics the antibiotic's going to try to poke holes in that peptidoglycan cell wall so that then water will flow into that bacterial cell and pop it. And that's how a lot of your antibiotics work in your body. Archaea are also prokaryotic, no nucleus for that DNA, but their cell walls are made of something called glycoproteins, and this means that their cell walls have a sugar and a protein component, and that's different from bacteria. Their cell walls are just this type of sugar, this peptidoglycan. Now, archaea also have a different type of lipid in their plasma membrane, and these different lipids help the archaea to survive in very extreme environments. And what I mean by that is they live in places like hot springs and very salty places like the Dead Sea. So archaea, while they can be found everywhere, they have an affinity. They really like to be in these really extreme hot temperatures or high pHs or low pHs, and really salty environments is where you can find a lot of archaea, and you might not find anything else living in that environment besides archaea. So remember that the plasma membrane is just what's going to be separating the inside and the outside of the cell, and it allows certain materials to go through. We talked about that in the homeostasis and transport lessons. You can go back and review that if you need to. We also have the domain eukarya, and this is going to be all of your multicellular organisms and a few unicellular organisms. The DNA here is going to be contained within a nucleus. So you are a eukaryote because you have a nucleus, and we looked at that when we looked at cheek cells. Eukarya are divided into 4 different groups or kingdoms, and those are going to be your plants, your animals, your protists, and your fungi. And we'll look at that here in just a few minutes as well. But I'm going to back up for just a minute. Remember, we talked about these protists here, or these 3 unicellular organisms that you need to know that are not bacteria. So they do have a nucleus, and these are some examples of unicellular organisms that would belong to domain eukarya. So when we look at these different domains, we can arrange them on something called a phylogenetic tree, okay? And a phylogenetic tree is something that's going to show evolutionary relationships between these organisms. So we assume that originally we had a cell, and that cell reproduces until we get different populations of that cell, all of the same type of organisms living together. Well, one type of population might have a mutation that allows it to survive a little bit better in its environment. And so that mutation is going to be passed on because those organisms are better able to survive and reproduce. So eventually, those populations are going to accumulate those traits, and we're going to have a split. And once we have this split into populations, the species are going to be formed, or in this case, the domains. These organisms are going to be too different to reproduce with one another. So here we have our common ancestor reproducing, different populations start to appear. And then one of these populations is going to remain similar to what it already was, with it might mutate over time. But we're going to have that unicellular organism, peptidoglycan in its cell wall, no nucleus, very primitive, and these are going to be your bacteria. The other population developed these glycoproteins in the cell wall and some other unique adaptations that helped it to survive in these really crazy environments. And so this population developed into the archaeans and the eukaryotes. So as we go along, we had the primitive cells, much like the archaea, and here we're going to have another split. These populations are going to develop different characteristics. The archaea retained those primitive characteristics of not having a nucleus, but the eukaryotes were better able to survive, and that trait got passed on. And so we ended up with this whole group of eukarya. And so these are the 3 domains, and every organism is going to belong in these 3 domains. And again, this structure is called a phylogenetic tree. We're going to look at these a lot in biology. So here are your 4 kingdoms of eukarya. We have plants, animals, fungi, and protists, and we're focusing on the protists today. But here's just some characteristics of these different kingdoms. Plants are producers or autotrophs. They make their own carbohydrates using energy from the sun and photosynthesis. They do have cell walls, and their cell walls are going to be made of cellulose. And they use that chlorophyll for photosynthesis. They have this very large vacuole. These are some characteristics that set plants apart from these other kingdoms. Animals are heterotrophs. They cannot perform photosynthesis. They have to go out and consume those carbohydrates that something else has made or eaten. They do not have cell walls. Cell walls help organisms stay tough and rigid, and animals don't need that. They need to be able to move. Animals do not have chlorophyll because they do not use photosynthesis, and their vacuoles, if present, are very small. Fungi are also heterotrophic, but these are decomposers. They're going to take that material in dead organisms and break it down and recycle those nutrients back to the environment. They also have no chlorophyll, but they do have cell walls. So they have some characteristics that you're going to notice between plants and animals. Protists are very diverse, and they contain characteristics that make them very difficult to put in any of these other eukaryotic groups. Some of them are unicellular, some of them are multicellular. Some of them can do photosynthesis and be heterotrophic, and some of them are only heterotrophic. They use a lot of different ways to move around, and we're going to be looking at that here in just a few minutes. So again, we have this phylogenetic tree. And this phylogenetic tree is showing you the different kingdoms of eukarya. And at some point, the protists are considered to be the original kingdom because they are so diverse and their characteristics could have given rise to all of the things that we see in the other kingdoms. So we have two different types of protists, but I just want you to think of them as protists. We have our plants, and at some point we had the split between the protists and the plants. They developed their own line. And over time, we had another split here between the animals and the fungi, and they developed their own characteristics in each of these groups. And we ended up with a lot of diversity, giving us all the different species that we have in these kingdoms. And we'll talk about that more later in the semester. So phylogenetic trees show ancestry. And remember, the further down we are on the phylogenetic tree, the more primitive that organism's characteristics are going to be. And when we see a split that's later on in the tree, all the organisms from that split evolved more recently. Now, to study unicellular organisms, we use microscopes. Most unicellular organisms are too small to be seen without a microscope. So these are tools that scientists use to increase the size of an organism, make it appear larger, and to be able to see that organism in more detail. Now, in class, we've gone over all of the parts of a microscope. We talked about this in your lab that I recorded for you as well. So the biggest thing I want you to know about a microscope is, of course, how to focus one. And if you're in class with me, then you'll learn how to focus a microscope. If not, then make sure that you watch your lab and you understand how that focus is going to work. But also how to determine your total power of magnification. Now, we have a lens that's in the eyepiece, and we have a lens that's in each of these objectives. So we have to multiply the power of the objective times the power of the lens in the eyepiece. So for example, all of the microscopes in this school have an eyepiece that has 10 times magnification. That means if you look through just the eyepiece, something's going to be 10 times larger than it normally is. The objectives on our microscopes are 4, 10, and 43. So the objectives alone can magnify something to be 4 times, 10 times, or 43 times larger than it normally is. But if we want to find our total power of magnification, we have to remember that we're looking through 2 lenses, and so we're going to multiply the powers of those lenses together. So for example, your eyepiece in the school would be worth 10, and there are microscopes that have different types of eyepieces. And if you're looking at low power, then you're also looking through a lens that's worth 4. So on low power, we would multiply 4 times 10, and that means something is going to be 40 times larger than normal with your naked eye. If we are looking through the medium power, then you would look through 10 and 10, and that's going to give you 100. So on medium power, something's 100 times larger than normal. And if we are on the high power, 10 times 43 is 430. So the maximum power that you're going to have is going to magnify something to be 430 times larger than it would be with your naked eye. We're going to look at some unicellular adaptations, how these single-celled organisms have certain structures or behaviors that help them to survive and reproduce, and that's what an adaptation is. So if we have an adaptation that is heritable, it can be passed on to offspring, then if it helps an organism to survive longer in its environment and reproduce more, then we should see more and more offspring that have that characteristic. So it's going to build up in populations over time, and that's how evolution takes place. So we have some special unicellular adaptations we're going to look at. Flagella, which are these long tails that you see on the bacteria and this Euglena here. And these long tails are different between eukaryotes and prokaryotes. They're constructed differently, but we're not going to worry about that for the sake of this class. We have cilia, which are these little tiny hairs that we see outside of this paramecium. Pseudopods, which we are seeing here on this amoeba, these are extensions of the cell membrane that help the cell to move along and gather food. We have contractile vacuoles, which are these star-shaped organelles that you see here. These are common in protists that live in ponds, and we'll talk about why that is. Eye spots, which do exactly what they sound like they do. They're this little spot that helps an organism to detect light. Now, they can't see in detail, but we'll get more into that in a minute. And then we have taxis, and taxis is the plural of this word. And a taxis is a behavior that an organism has that's going to help it to survive in its environment. Cilia are short little structures. They look like hairs, and we find them all around the outside of an organism that has them. So they're very common in paramecium, and this is a diagram of a paramecium here. You're also able to see a paramecium here. This is a real one that was taken with an electron microscope, and we see the little cilia here as well. So what these are for is movement primarily. So they flip around back and forth, and they're going to help this paramecium to swim through the water. And that's where most of our protists are going to live, is in water. Now, these little cilia can also be used like a broom to sweep food into this oral groove and gullet that the paramecium is going to use as its mouth. Okay. Now, cilia are also found in multicellular organisms like us. We find them in our respiratory tract, in our trachea. And those cilia that we have are there to help beat things out of your lungs that you don't want in there. For example, if you get sick and you start coughing up a lot of mucus, those cilia are beating that mucus up and out of your lungs. If you smoke, then the tar and the toxins in that cigarette smoke can actually destroy those cilia and make them to where they can't move and they can't function. So that's where a lot of diseases associated with smoking are going to start and where they're going to come from. So big deal, cilia are used in movement and feeding. Flagella are similar to cilia, but they usually aren't present in such great numbers. They're also longer. So this is an example of a Euglena, a protist, that has a flagella, and you can see it's a very long whip-like structure, and they can whip back and forth to move the cell, or they can spin like a propeller, depending on the type of organism that you have. Now, some organisms only have one flagella, like this Euglena, but some organisms have a lot of flagella, like this bacteria here. This is an E. coli cell, and you can see all of the flagella sticking off all around it, but they're a lot longer than cilia would be. They are used for movement, but they are not used for gathering food, so that makes them a little different than cilia as well. The only cell in the human body that uses a flagella is the sperm cell, and the flagella helps the sperm to swim to the egg. The word pseudopod translates to false foot, pseudo for false and pod for foot. This is a temporary projection in a cell that's going to help it to move, and we see these in amoeba. So this is an example of an amoeba here, and this little projections that you see, each one is called a pseudopod. Pseudopodia is just plural. Now, the way these work, the amoeba, when it wants to move, is going to build cytoskeleton in the direction it wants to move, and that's going to stretch out the cell membrane, and cytoplasm is going to flow into that. So it's going to stretch out a pseudopod, and that pseudopod is going to link to a surface. And then the pseudopods that are behind are going to let go, and that's all that cytoplasm is going to flow back into the cell, and then they can stretch their membrane out forward and grab onto something else. So they crawl along using these pseudopods. And these pseudopods are very flexible, so they can actually wrap around a material and pull it into the cell. So you can think about it like it's enclosing something and then taking that in. So This process where a pseudopod is going to wrap around something and bring it into the cell is called endocytosis. More particularly, it's called phagocytosis. And pseudopods are also used by a cell in our bodies, the white blood cell. And these pseudopods help the white blood cell to engulf pathogens, to engulf bacteria and viruses that would make us sick, and they can crawl along using this amoeba movement. A vacuole, remember, was an organelle, especially in plants, that stored things like water and waste and nutrients and all that good stuff. Now, in protists, we have something called a contractile vacuole, and it also stores water, but just water, and it is able to contract. So remember hypotonic solutions. If a cell is in pure water, that water is going to want to flow into the cell. And if too much water enters, then the cell can burst. So these contractile vacuoles help keep that from happening. They regulate the water balance and help the cell stay in homeostasis. So this is a contractile vacuole here in a paramecium. And as the cell fills up with water, the contractile vacuole is going to swell up like this. And then all of a sudden it's going to squeeze all the water out. And this is one that's starting to squeeze, and they take on this star shape when they empty out. So the cell will burst without these because, again, all that water is flowing into the cell from a high to a low concentration. This is a video that will show you how these contractile vacuoles work. So here's the contractile vacuole. You can also see the little cilia beating, and you can see how the oral groove and the gullet are taking in food. It's a really cool video. It's very short, so find this on your PowerPoint and check it out. It's really cool. Eye spots do exactly what they sound like they do. They detect light, but that's all they can detect. They can't see detail like our eyes can. So Euglena are little protists that are going to have these eye spots. And a Euglena is a photosynthetic protist. It's got these chlorophyll molecules in it, in these chloroplasts. And remember that chloroplasts and chlorophyll are responsible for processing light and converting that energy into carbohydrates. So if a Euglena is going to use photosynthesis, then it needs to be able to tell where light is. So this is an example of how organelles can work together. The eye spot can detect the light, the flagella can swim toward the light, and then the chloroplast can process that light and use the energy in the light to make its carbohydrates out of carbon dioxide and water. So Euglena are unique because they can produce their own food, but they can also consume food. And again, they have this little flagella that helps them to move. And you'll see this is a Euglena here. There is its eye spot. And it also can wiggle around to kind of help with its direction. Taxis, or taxis for plural, are involuntary responses, behaviors that organisms are going to have to respond to what's going on in their environment. And we call those things that elicit the response, that cause the response, stimuli. So a taxis helps organisms to respond to stimuli. And a taxis can be positive or negative. A positive taxis is going to cause organisms to go toward the stimulus, and a negative taxis is going to cause organisms to go away from the stimulus. And these are some things that organisms might respond to. Chemicals, for example, if I smell the chemical in the air from frying bacon, I'm going to go toward that stimulus because I'm going to want to get me some of that bacon. If I were to smell smoke in my house, though, I'm going to want to get out of there because don't want to be in a house fire. So that would be a negative taxis. Light, you can think about plants growing toward a window. You can think about bats that don't come out during the day. They have these responses to light. Positive, going toward that window, and away, the bat hiding from the light during the day. With magnetism, think about migration. Birds use these magnetic fields in the planet to help them navigate back and forth through the summer and winter months. And then electric current, you can think about sharks and how they use electricity in the water to tell where a living thing is that they might want to eat. So chemotaxis and phototaxis are two things that we want to talk about, and we'll look at those here in just a minute. But here's an example of taxis. We have this solution with these little bacteria in it. The white dots represent the bacteria. And this is a tube that has nothing in it, nothing that's going to cause these bacteria to respond. So they stay pretty spread out in this solution. We also have in this second tube a attractant, something that these bacteria might want to eat, for example. And we notice they want to congregate around the tube. So this would be a positive taxis. Here we have a tube that has a repellent in it. So something like an acid maybe that these bacteria don't want to be around. So they're going to get as far away from that as they possibly can. This would be a negative taxis. And down here, you're just seeing a diagram of how bacteria move. They use something called a run and tumble. So in a control, they're just moving around randomly. The bacteria is going to run in a random direction, and then it's going to stop, and it's going to tumble around for a minute. It's going to take off in another random direction. When there's something that's attracting them, then they're going to run. And if they detect they're not going toward it, they're going to stop, they're going to tumble, and they're going to run again. If they're still not going toward it, they're going to stop and tumble, and they're going to take off in a random direction until finally they get to the right direction that's taking them where they want to go. If this is a negative taxis or something that's causing these bacteria to go away from them, then they're going to do the opposite. They're going to run in a random direction. If they detect they're going toward that thing they want to get away from, they're going to tumble, and they're going to go in another random direction until finally they're moving in the direction they want to go. This is called the drunken walk because it is so random, and these bacteria have a hard time controlling the direction that they're actually moving. So chemotaxis and phototaxis. Chemotaxis would be an example like we just looked at with the bacteria. Also, so we have chemotaxis and phototaxis. Chemotaxis is movement toward or away from a chemical. We just looked at an example with that with our bacteria. And I gave you a couple of examples like smelling bacon. It's also hunting season here. So a lot of you may hunt deer, and maybe you use pheromones to attract deer to come into your hunting ground. So that would be an example of a positive taxis that would bring those deer in. A phototaxis is movement toward or away from light. Photo means light. So you have an example here of a moth going toward a light bulb. Cyanobacteria, these are bacteria that are capable of photosynthesis. They're going to want to move toward light for food production. Same thing with the Euglena that we looked at earlier. Malaria is a protist that is considered a human parasite. So it lives off of humans, and it's transmitted by a certain genus of mosquitoes. And we don't have that genus in America here. But these mosquitoes, when they bite you, they're not only sucking out your blood, a mosquito also inserts its saliva. So when you get that itch from a mosquito, it's a localized allergy that you're having to the saliva of the mosquito that bit you. So the transmission occurs when that saliva is put back into a human, and it's carrying this little protist with it. So it's inserted upon the bite, and then the protist is going to attach to a blood cell. You can see one of these little malaria here, and it's attached to a blood cell here. You can only see a corner of it. You can also see some of them in purple here, and these gray things are blood cells, so you can see some of the blood cells that have been affected by these little protists. And again, these are not nuclei. Actually, blood cells in humans do not have nuclei. So any little purple thing you see inside of that blood cell is a malaria. The blood is going to carry this protist to the liver where it's going to reproduce. And this is where the infection is going to get nasty. So malaria kills about 500,000 people a year. That's quite a bit, and infects almost 250 million. These red areas that you see here are where the malaria parasite is really common. So if you go to these areas, you need to be careful about watching your body when you come back or while you're there to make sure that you don't develop any symptoms to this disease. People that carry sickle cell disease, they carry it but don't necessarily have it, actually are a little resistant to this disease. And we'll talk about sickle cell a little bit later. But this particular shape of the blood cell that you have during sickle cell anemia makes this blood cell where it cannot possibly take in these little protists. And so these people don't typically get malaria. And that's why the sickle cell gene is still around today, supposedly. Here's some resources I used. Hopefully you enjoyed this lecture. I think it's really neat to look at unicellular organisms. So please make sure that you complete your assignments and take your quiz. And this will be a very short unit, so the test will be coming pretty quickly. See you next time.